today we're going to have a discussion about a very important topic, cell-mediated immunity. And cell-mediated immunity is part of the adaptive immune system. It's a very important part. And we're going to examine the different cells that are activated in this response. We're also going to talk about the different proteins and cytokines and chemokines that are secreted by these cells. And then we're going to bring it all together and discuss basically the entirety of the immune system in relationship to how all of these cells interact. So, as you can see, the adaptive immune system is composed of the humoral immune response, which we previously discussed, and it involves the B cells, the plasma cells, and the antibodies. But today we're actually going to concentrate on the cellular immune response. And really, the cellular immune response is all about the maturation and activation of the T cells. So the T cells, as you can see, also further differentiate into three pathways, Th1, Th2, and Th17. They also involve the dendritic cells, and all of these cells interact together in relationship to killing the infected cell and also activating other cells that are involved in immunity. So today in particular, we're going to concentrate on the various hallmarks of cell-mediated immunity. So these hallmarks include, as the central player, the lymphocyte, the T cell lymphocyte, and we're going to discuss what the T cell is, what it looks like, where it comes from, how it differentiates. We're also going to examine T cells in relationship to how they interact with antigen presentation. We're also going to talk about the different subsets of T cells, the different variants and types, and also the function and what they do. We're also going to talk about very much in detail the T cell receptor and its relationship to antigen presentation. We're going to include the discussion of secretion of cytokines and chemokines that occur with the T cells. And then in particular, we're going to talk about T cell regulation and how T cell regulation operates in terms of immune tolerance. And furthermore, we'll talk about the T lymphocyte as the primary actor in all of these activities and how the T cell is directed towards pathogens, in particular, that actually have survived the first innate immune reactions. We're also going to discuss the activation of phagocytes by the T cells and the antigen-specific T cells, and also how these antigen-specific T cells secrete cytokines. And again, all of these different players work together to eradicate various types of pathogens. So we're going to see the elegance of the T cells in relationship to how they very specifically identify these pathogens and work with many other cells to execute basically eradication of the pathogen, but also finally how the T cell is able to regulate itself, because obviously after the pathogen is gone, you want the immune system to become deactivated. So that's the really important part of the T regulator system. So in terms of the cell-mediated immunity, most importantly, these processes induce apoptosis in cells displaying epitopes of these foreign antigens. Because remember, especially viruses, but also bacteria. Now, if we have a host cell, right, with a nucleus, how on earth will the immune system know, for instance, that that particular cell has various viruses inside the cytoplasm? Okay, or even if that cell is for instance, also infected or independently infected with bacteria, how does the immune system recognize that indeed that particular cell is infected? It does so through this antigen presentation, because with antigen presentation, we actually get these antigen shapes that are expressed on the cell surface, and then by having those epitopes or those shapes of antigens expressed on the cell surface, the T cells can identify that indeed that is an infected cell, and then will go through various processes to eradicate that infected cell. Because if there are other normal cells, obviously the immune system, we don't want the immune system to eradicate normal cells. So these foreign antigens are expressed on the cell surface. Antigens from viruses, antigens from intracellular bacteria, and neoplastic cells, which are cancer cells that have tumor antigens on their cell surfaces as well. But not only does the T cell recognize these infected cells and eradicate these infected cells, but the T cells also activate other cells that also basically migrate to the scene and also work towards eradicating these cells as well. So, continuing our discussion, this will probably be the most complex lecture of all of them. Anyway, the T cells also stimulate cells to secrete these signaling molecules called cytokines, and these cytokines bring on these other cells that certainly help with this eradication process, and these T cells respond to intracellular antigens, and which in this case require the cell-to-cell direct contact through these cytokines that bring other cells on the scene, because these macrophages they don't have the specificity that T cells do. But when the macrophages see that the T cells are attacking these cells, will also participate in the process through the secretion of these cytokines. Okay, so let's examine the T cell in detail. So the T cells mature in the thymus. That's why they're called the T cells. They are characterized by a T cell receptor, and the T cell receptor is really the brain of the T cell interaction. And it's a very specific, attachment or receptor that is certainly, it's an integral protein, right? And it goes completely through the lipid bilayer of the plasma membrane. But most importantly, it has several components. One of the most important components of the T cell receptor is this major histocompatibility complex called MHC. Now, MHC is designed to alert the T cell to other cells in the body that belong to the host. And you'll see here, so here is an antigen-presenting cell, and that antigen-presenting cell, and we're going to go through the various cells that do present antigen, and that antigen-presenting cell also has a component, this MHC component, that is an integral protein and cell receptor. And right here, this peptide is the antigen. So the antigen-presenting cell has that major histocompatibility complex, and right at the top of that complex is the antigen. So that major histocompatibility complex of the antigen-presenting cell matches up with the antigen-presenting complex of the T cell receptor, and by that process, then the T cell receptor truly identifies that indeed this peptide is an antigen. And that appears to be a very complex process. It certainly is, but it almost virtually 100% guarantees that indeed this peptide is the true antigen. And then once the T cell identifies that peptide as the antigen, it will execute the machinery designed to identify the pathogen with the antigen and eradicate that pathogen. So this T cell will only act on an antigen if it's presented by an antigen-presenting cell. So if an antigen, for instance, is just floating around in the extracellular fluid, the T cell is not going to respond to that. It's only going to respond to the antigens in which are presented by these antigen-presenting cells. But we're going to see that potentially other cells can get involved in the process too. So let's talk more about the T cell. The T cell is a type of leukocyte, and it's also responsible for immune responses that lead to organ transplant rejection. So these three attributes, these three types of responses, are instances where the T cell can certainly become a little bit disordered. And for instance, allergic conditions, what happens there is the T cell responds to naturally occurring antigens that are not pathogens. So for instance, gluten antigens, or pollen, or bee stings, or peanuts. These are antigens that are not damaging to the host, but because in some cases the T cell is so hyper-responsive, it actually, again, identifies those particles, proteins and so forth, as actual antigens. But again, the T cell only reacts to these antigens if they're presented by the antigen-presenting cell. So in that regard, the T cell is really responding, of course, to other cells that have also identified this antigen as being maladaptive, when indeed it's not. It's not harmful at all. But we have other autoimmune disorders where we have immune reactions to the pancreatic beta cell, for instance, that leads to diabetes. We have multiple sclerosis, which is another autoimmune disease. We have rheumatoid arthritis, where the rheumatoid factor becomes the antigen, and then we have immune reactions against different joints in the body, synovial fluid, for instance. But the main function of the T cells is really to target intracellular pathogens. So we're going to continue to discuss this process. Okay, so how do the T cells maturate? Okay, so as we discussed, T cells are found in the thymus, and what they do is they remain in an immature state, and they stay there until they are activated in relationship to infection by a pathogen. So that process is called thymopoiesis, where we actually have T cells differentiating to then get launched and prepared for eradicating the pathogen. So the thymus conducts a testing process of both positive and negative selection. So as you can see in this figure down here, we have a very elaborate array of all these different types of receptors that basically, once an antigen is identified as an antigen, right, and it's presented to the T cell by an antigen-presenting cell, we get a self-selection of T cells that have configured themselves to have configured their T cell receptors to be very specific for that antigen. So then the particular type of receptor that's already available will get called up and selectively and basically selected for, whereas cells that react to the self, so where we have T cells that just have that major histocompatibility complex, those cells are triggered to undergo apoptosis. So basically we have a selection process where the T cells that carry the specific T cell receptor get selected for, and the T cells that don't have that receptor get selected against, or they actually undergo apoptosis, which is another word for cell death. So to continue our discussion about T cell maturation, you might then ask, well, how on earth do all of these T cells get created? So for each unique infection by each unique antigen, right, we launch a unique population of T cells. So in order to create those unique T cell populations, we undergo somatic gene rearrangement, where not the inherent, well, I mean, all the genes and all the cells in the human body come from the germline DNA, but in this particular case, the particular situation of that specified T cell receptor is what generates the particular configuration of these somatic genes that then go on through mitosis, right, to create more T cells that have that very specific T cell receptor. So as we can see from this figure down below, that there are an infinite number of configurations that are possible in terms of creating this very specific T cell receptor, and therefore these T cell receptors can of course get selected for through this somatic cell gene rearrangement. So then now let's talk about another attribute of T cells, and they go through what's called this cluster of differentiation. So they basically are differentiating in clusters. So here we have the CD34 immune stem cell, and that stem cell can become several different kinds of immune cells. So the granulocytes, the monocytes, the T lymphocytes, the B lymphocytes, and the thrombocyte. So in the case of the T cells, right, we have a development of this very specific population of T cells that are configured to be very specific to whatever pathogen is infecting the human host. But also there are very specific proteins that are expressed on these different immune cell populations, and these proteins can act as receptors, these proteins can act as transporters, and they can also act as ion channels. And of course these cells all have a plasma membrane, and so what can happen is we can have this very specific population of T cells developing, but we can also have a very specific population of B cells developing as well. So then we get what's called an immunophenotyping, right, where we get a particular class of cells developing in relationship to particular infections by these different pathogens. And we can define the cells by the molecules expressed on their surface. And these molecules can indicate these cell-specific immune functions. So in this case, and we'll see on the next slide too, how that process works. So in terms of the nomenclature, the way you describe these cells is, for instance, a CD45 plus, CD31 negative means that here in this T lymphocyte we have a CD45 being expressed, and a CD31 being regressed or not expressed. And also we can have for instance, a CD45 where that particular protein is expressed, and a CD3 where that particular protein is expressed. So we can see that here in the cell. Here we have a CD45 expressed and a CD3 expressed. So that's really a way that you can classify these different T cells in relationship to the type of immune response elicited by these different antigens. And so if you certainly look at the immune literature or even look at lab results, or if you work in any part of immunology, for instance, you'll see this terminology used quite frequently. So now that we've discussed in detail the maturation and development of the T cell, let's talk about antigen presentation, because antigen presentation is an integral principle of the adaptive immune system. And again, this T cell is very particular. It is not going to react to an antigen unless it's presented by an antigen-presenting cell. So this antigen presentation involves the interaction of the surface antigen with the T cell receptor. So here we have these different types of presentation. We have a direct presentation, we have a cross presentation, and then we have what's called a cross-dressing. So let's just go through those processes so you can get a better handle on how that works. So a direct presentation is where we have a dendritic cell. Now remember, a dendritic cell is a cell found in the immune system. It's one of the presenting cells. And that dendritic cell has at the end of its major histocompatibility complex right here, it has the antigen. And so then the major histocompatibility complex receptor of the T cell then recognizes that this antigen is being presented and then is activated in relationship to developing a T cell receptor. So that's direct presentation. Now cross presentation is where we have an infected cell, right? And then those infected particles pass over to a dendritic cell that then takes those infected particles that represent the antigen of the pathogen, and then goes through the presentation process to the T cell. And now the cross-dressing here is kind of a double event, basically, where we have the infected dendritic cell that then actually passes over to a healthy dendritic cell, the antigen, and then the antigen is presented by the healthy dendritic cell. So there are certain cells that are dedicated, and those are considered professional. They're dedicated to presentation of antigens, and these cells include the dendritic cells, the B cells, and the macrophages. Now, these three cells also engage in other processes, but in relationship to the T cells, these cells are always recognized as antigen-presenting cells. However, we can have other cells present antigen as well. Now, glial cells, there's a particular class of glial cells in the brain that function in immunity. Those are called microglia. So let me see if I can write that here. Glia. Okay, and those are glial cells that are devoted to immune responses, and they actually migrate freely in the cerebrospinal fluid, and one of the things they can do is actually present antigen. But we also have other cells. We have thymic cells. Okay, now that makes sense because of the thymus is where T cells mature. But also thyroid cells. Now, these thyroid epithelial cells these follicles that exist in the thyroid. Now there's a particular kind of thyroiditis. It's called Hashimoto's disease, which is an actual autoimmune disease of the thyroid that is certainly facilitated by these T cells. But we also have vascular endothelial cells that can also present antigen, and that may prove to be a problem in relationship to atherosclerosis. So to continue with our antigen presentation, so this contact between an antigen-presenting cell and the T cell receptor stimulates important signaling events. And so here is actually each of these different types of presentation are described in detail in text. So we have the direct presentation, and then we have cross presentation, and then finally we have the final, which is facilitated presentation. So so that completes our discussion of antigen presentation. Now let's focus on the different types of T cells, the different subsets of T cells, because these different subsets actually execute different functions. So there are 3 pathways. Okay, there's Th1, Th2, and Th17. And the Th17 has just newly been discovered in the last several years. But the Th1 and Th2 pathways have been known for quite some time. So again, it's important to remember that these cells, again, are all driven by this antigen presentation. So once we have that event occurring, we can activate each of these 3 pathways depending on the function that is required. So the Th1 pathway, that is the pathway that is very specific to eradicating intracellular pathogens. Okay, now these pathogens can be both bacteria, they can be viruses, and again, they can also be antigens that are found on tumors. So we'll go through that in detail, but also the Th1 pathway secretes, or the Th1 cells secrete interferon gamma, and that interferon gamma actually attracts macrophages to the infection site. So these Th1 cells, Th1 T cells, T helper 1 cells, they produce cytokines, and they also activate these cytotoxic T cells that are designed to eradicate these pathogen-infected cells. Okay, now remember, these cells are host cells that are infected. And in order to eradicate the pathogen, you need to eradicate the cells that contain the pathogen. So the Th1 pathway actually bridges the adaptive immune system, the cell-mediated immune system, and also the innate immune system. We can never forget about the innate immune system because it's always there too. Okay, so now Th2, which is this pathway here, now Th2 is actually a pathway that facilitates B cells, right? And remember, B cells produce antibodies. Okay, so how the Th2 T cells, I guess, elicit the action of the B cells is through secretion of IL-4, which is an abbreviation for interleukin-4, and it stimulates these B cells to produce antibodies. Now remember, the antibodies have their own way of being specifically designing shapes that fit in a complementary way with the antigen. And we've described the B cells in a completely separate lecture. Now, the final pathway is the Th17 pathway, and that pathway activates neutrophils. Now remember, neutrophils were the main cells that were found in the innate immune system, and they were also one of the first cells on the scene in relationship to the beginnings of invasion by the pathogen and infection. But now, here at this later stage of immune response, the Th17 pathway also activates these neutrophils, and it does so through secretion of IL-17 and IL-22. So remember that the neutrophils are important because they cause or create inflammation, and they change the mucosal and the epithelial barrier. They stimulate production of these antimicrobial proteins. Remember the complement pathway, okay? We also get complement here as well. So it's almost like a later stage feedback mechanism to bring the innate immune system back into the fold. Now, if you have a deficiency in Th17, you can also then open up the gate to being more susceptible to opportunistic infection, for instance, and the Th17 pathway is also associated with autoimmunity. Now, the CD8 cells are the cytotoxic killer T cells, okay? Those cells are found in Th1, and those cells are the primary defense cells against tumor cells and against viruses. These cytotoxic T cells target cells with antigens. Now, most especially with these cells that are infected with viruses, right? How would you know that a virus was infecting, for instance, some respiratory mucosal cells and not others, right? So you want to eradicate the host cells that are infected with the virus. Well, guess what? That's what the cytotoxic T cell does, and it does this through the T cell receptor again, right? That's attached along with the major histocompatibility complex type 1, and also these cytotoxic killer cells kill parasites, they kill worms, right? Now worms are, another class of organisms, right? And we will discuss them later on in the class or in the course. So now you might say, well, what other weapons, right, do the T cells have? Well, they have some other ones too. Okay, so some of these infected cells undergo apoptosis by not just being engulfed and killed, right, but there are these two types of molecules that can be released by the cytotoxic killer T cells, and these molecules are called perforin and granzyme. Now, perforin was also a type of mechanism that was utilized in the complement system. Okay, so the perforins actually, it's almost like they're little bombs, right? They're proteins that create holes in the cell membrane, and what ends up happening is, because the concentration of the cell is much higher or much more concentrated than the extracellular fluid, you get a rush of fluid inside of the cell, and then you get a bursting of the cell. And these granzymes are actually particular enzymes that induce apoptosis of these cells. So remember, the Th1 pathway executes its action through these cytotoxic T cells, which secrete perforins and granzymes, and then also secrete this cytokine, right, that goes ahead and calls up the macrophages that then come along and engulf and phagocytize the cell. So it really has three weapons to its advantage, right, that can be utilized in relationship to eradicating these infected host cells. Now, now the question is, well, what happens if, for instance, the T cells identify normal host cells? Okay, and if that happens, then obviously you want to suppress this activity. So, for instance, when an individual gets an organ transplant, generally what happens is a close relative of the person is asked to donate organ if possible, or a big call is made to all over the world through these various databases to bring in, for instance, bone marrow or other organs or other or blood products, so to speak. And so when you bring in an organ, for instance, the immune system recognizes that organ transplant as being foreign tissue and launches immune actions against that tissue. However, if you want the transplant to hold, if you want the transplant to take, you need to suppress this cytotoxic killer T cell response. And so that's why we have developed these immune suppression drugs to actually mute that process. So for instance, an organ transplant can hold. We also use these pharmaceuticals for allergic reactions to antigens like pollens or food, peanuts, or bee stings, and also various autoimmune disorders. Now, we're going to have a separate in-depth discussion about that, but it's important to also mention it here because the T cell response can actually also be maladaptive. So now let's talk about regulatory T cells. These T cells are basically just as important as these cytotoxic killer cells, right? Because once the infection is over, once the pathogen is eradicated, you want to be able to turn off the immune response. So these regulatory T cells function to do that, but also they have other really important functions as well. So this regulatory T cells suppress activation of the immune system to prevent autoactivation and autopathology. So the regulatory T cell system can actually suppress allergic reaction, can suppress autoactivation on its own or autopathology. So for instance, you don't want the immune system to attack the joints in terms of, the rheumatoid factor. You don't want an immune reaction against those things. Now obviously, the immune reaction can escape the regulatory system, but potentially if the regulatory system can keep these things in check, then for instance, you won't necessarily require any pharmaceutical intervention. So these proposed functions that so the regulatory T cells maintain self-tolerance. So really what you want the body to be able to do is to tolerate itself, to maintain a commensal relationship with all the other cells in the body so that we don't really get this immune activation in places where we don't want immune activation. You also want the regulatory T cell system to downregulate responses to allergens like asthma, for instance. Okay, so you want to be able to suppress and downregulate responses to allergens. So one of the things we know, for instance, is sometimes asthma attacks occur when the regulatory T cell system is suppressed. Okay, and sometimes that can happen even with stress, for instance. Okay, so then we have other types of immune tolerances. We have commensal bacteria. So you might say, well, what about the bacteria in the gut that are friendly bacteria? What about the bacteria that are there that even metabolize important nutrients? So there are a particular strain of bacteria, for instance, that live in the colon that synthesize vitamin K. So if, for instance, you take a long-term antibiotic for some other infection, what ends up happening is you eradicate the bacteria found in the gut, and then, for instance, you can require vitamin K supplementation because you become deficient because the bacteria that produce vitamin K are eradicated along with the infectious bacteria. So it's important that the regulatory T cell system regulate and suppress the cytotoxic T system in relationship to allowing the commensal bacteria to exist. Also, we want to enable the T cell regulatory T cells to support the transplanted tissues. You want the regulatory T cell system to suppress the mother's reaction to fetal antigens. So an example, for instance, are the fetal thyroid hormones, that you don't want the mother to react to those hormones in terms of you don't want any kind of immune reaction. Well, how does that happen? That happens through this regulatory T cell system. Now, what about tumors? Now, unfortunately, one of the reasons why tumors take hold is because the regulatory T cell system sees those tumors as being part of the host. And so it suppresses the potential T killer cell activity in relationship to potentially eradicating the tumor. So in a sense, the regulatory T cell system can overreact and suppress immune actions that are actually beneficial. So these are different types of cytokines, okay, and they're released by different kinds of cells. So we have, as we've already discussed, we have interleukins, we have interferons, and then we have growth factors. So you can see in various cells in this diagram different interleukins, different growth factors released, and different interferon. We have interferon gamma, and we have interleukin, all these different types of interleukins. And these are all really important in relationship to signaling and communicating in relationship to launching these immune actions against pathogens. So the chemical messengers are really also known as cytokines, and these are proteins that are released by the immune cells for communication and signaling. They can both elicit immune response, and they can also turn off immune response. So here's another picture, the same picture again, and down below we have a table here that can illustrate all of these different interleukins and cytokines and so forth that cause these different reactions. So we have Th1 cytokines. So first of all, we have lymphocyte growth factors, and these growth factors are responsible for clonal expansion of these very specific T cells, and they also influence the 3 pathways, and examples are different interleukins. We have Th1 cytokines that elicit the Th1 response, and these are clonal expansions of those cytotoxic T cells, and those are interferons as well as interleukins. We have Th2 cytokines, and remember, those elicit the B cells, and those increase antibody production, and there are various different types of interleukins there. And then we have Th17 cytokines, and the Th17 cytokines, remember, are engaged in regulation and also autoimmunity. So they turn on these other interleukins, interleukin 17, for instance, and also interferon gamma. We have pro-inflammatory cytokines that are released in the presence of inflammation, and they elicit innate immune responses, and the innate immune system can also certainly secrete those pro-inflammatory cytokines. And then we have anti-inflammatory cytokines that function in relationship to regulation, and they decrease the lethality, they decrease the occurrence of autoimmune diseases, they decrease the action of inflammatory disease or inflammatory genes, and so forth. So really, this illustrates the complexity of all of these different cells and the myriad ways that they interact. But the thing about it is, in terms of trying to really manage the information, if you can look at and study the immune system in relationship to these processes separate from one another, and then bring them all together, it's far easier to understand them that way than to start out studying them in relationship to how everything works together in the beginning, because then it's very difficult to manage all the different types of communication and different activations and different deactivations, which things go through apoptosis, which cells get activated. It's quite challenging to manage it all, but if you're very methodical about your studying and characterizing each of these processes, by the end, you'll actually really understand immunity at a very deep level. And this study of immunity, this immunology discipline, is integral to really understanding understanding microbiology. So, what about chemokines? What are chemokines? Chemokines are chemicals that are released by these T cells that cause other cells to be attracted. So they induce chemotaxis of other immune cells, like the neutrophils, the lymphocytes, the monocytes, the eosinophils, and other cells. Now, those cells also engage in very, and the innate cells certainly engage in activities that are a little bit more generalized, but the T cells and B cells, again, are very specific. And so when you release these chemokines, you draw all of these cells basically to the scene of infection, and then they can, participate in all of them together collectively, eradicating pathogens. So you can see that, for instance, in a wound that becomes infected, because a wound that becomes infected basically, becomes reddened, becomes inflamed, and then all of these different immune cells come onto the scene, and you can actually see that with wounds in terms of, an abscess or pus or all of these things. These are all immune cells that have attracted themselves to the scene of the infection through this process called chemotaxis. So there are three functions of chemokines. What do? They launch inflammation. They activate leukocytes to a wound for immune response and healing. They're homeostatic, so they operate in relationship to immune surveillance, hematopoiesis, antigenic surveillance in lymphoid tissue, and they also can launch an antiviral response. So they have various letter type designations, okay, and they really are meant to indicate the chemokines in relationship to how they function. So, my God, can you believe it? All right, so I'm not going to go through all of these things in detail. I think you can use this slide, this last slide here, to really allow yourself to fully understand how all of these cells interact and how they all work together in relationship to eradicating pathogens. So you can see, for instance, that some of these cells have receptors, and they have very specific antigens that are presented. And so here you can see at this level of the figure, right, we basically have the adaptive immune cells. And up here, right, to a large part, we have innate immune cells, but we also have other cells too. We have endothelial cells. We also have T memory cells. So just like the B cells have memory, well, the T cells have memory too. Some of the T cells stay behind with their very specified T cell receptors. And, you know, we have these other dendritic cells, we have plasma cells, plasma B cells, we have macrophages. Now, tumor cells kind of function in a similar way because they actually have their own antigens, right? And so forth. So these are just ways, so you can kind of have this slide here to be able to go through at the very end of your preparation for the immunology part of your examinations and really try to get a handle on what these cells all do. So, my God, let's ask ourselves some questions. So, what is the importance of the T regulator cells? So the T regulator cells, in my opinion, are just as important as the T helper cells. Okay, or the Th1 cells and the Th2 cells, because they enable the commensal bacteria to live in the gut. They suppress these autoimmune processes. They can potentially suppress transplant rejection. And also, most importantly, they cause the immune system to stop attacking cells when indeed the pathogen is eradicated. So the T regulatory system is a very, very important arm of immunity, and it's an aspect of immunity that is really becoming, there are more things being revealed about how important this arm of the immune system really is. So the second question now, what is the difference between a cytokine and a chemokine? Well, we just had a discussion about this, right? So the cytokine really is a signaling molecule. Cytokines are generally proteins, okay, and they serve a signaling function. So they cause cells to either become activated or deactivated. Chemokines, remember, chemokines are the cells that actually are attractants, right? So when chemokines are secreted at the site of infection, right, what that chemokine does is it's a call to action, right? It's a call to action for all these other immune cells. Okay, so it says this is where the infection is, and we are calling you to action. Okay, so a lot of the innate immune cells, which, remember, the innate immune cells are not specific, right? They're generalized. However, if these innate immune cells see that there's T cells and B cells that have specifically identified that site and those cells as being infected, then they're going to come on the scene and eradicate the cells as well. So these two processes are very important functions of these T cells and of cell-mediated immunity, because it's that mediation part of cell-mediated immunity that occurs through the action of these cytokines and these chemokines. Now, the final the final question has to do with what are the three distinct T helper cells pathways. So we have Th1, we have Th2, and we have Th17. Okay, now in terms of studying for your exam, I highly recommend that you take a blank piece of paper, right, and then trace down all the different things that occur in the Th1 pathway. So remember, the Th1 pathway is the pathogen eradication pathway. Okay, pathogen eradication, right? But what is the biggest part of that pathway? Number one, right? Antigen presentation. Okay, antigen presentation and major histocompatibility complex, right? All right, that's really important. And once the T cells identify what that pathogen is all about, then we get pathogen eradication. Now, what about the Th2 path? Remember, that's a series of steps where the T cells interact with the B cells and the plasma cells to create antibodies, or to synthesize antibodies. And remember, the antibodies are also very specific to the antigens. Okay, so we get antibody production. Now, the Th17, that pathway has to do, remember, with regulation. Okay, big time, regulation. So, this is another way that you can use to really characterize and fully understand how these T cells operate, because again, it's an integral part of immunity. So that's all we have for today. Thank you very much for visiting educator.com.